Okay, right. Assalamualaikum. Okay, very good morning to all students. Okay, so make sure you have what we call MIC 180 laboratory guideline. All right. Okay, we go for straightly for the ODL, fully ODL 100%. So ada dua version kat sini. Scheme of work of laboratory. Number one will be your version face-to-face plus ODL. Another one will be the fully ODL. So please use version 100% fully ODL. Okay. So kita akan ada face-to-face. Alright, so for today, we will learn about lab number 2. Alright. So what is lab number 2? Qualitative test for amino acids. Alright. So please bear in mind, okay, laboratory educations, all laboratory topics except lab number 4, which is colorimetric test for carbohydrates, okay, will be applied. Okay. So semua ni dia apply pada semua lab kecuali lab nombor 4. Alright, kenapa lab nombor 4? So tolong baca ayat yang ke bawah ni, assessment. So watch the video, okay, of principle of all labs provided. Okay. So please answer the short question to the test student understanding. So saya akan bagi setiap macam lab pertama tadi sebelum ni kita ada post lab question. So kena jawab within 2 hours of our lab session, right? Dan hantar pada saya. So the complete quiz is considered attendance for the lab session that weeks. Okay, so bagi saya, kalau awak hantar pada minggu tu, itu dikira sebagai markah, right? Okay, assessment will be only for lab number 4. Okay, so what is lab number 4? Colorimetry test for carbohydrate. So what you have to do to submit an individual report after watching the video for this lab. Okay, it means that only lab 4 will be your marks for lab report. Okay, so please bear in mind, for your lab report, 20% yang awak akan dapat. Okay, so total will be 20 marks in your course assessment. Alright, so maknanya 20 markah kat mana? Dekat dalam situ. So lagi 80 daripada test dan juga daripada assignment dan sebagainya. Alright. Completed lab report is considered attendance for the lab session that week. Alright. So maknanya dalam lab 4 nanti takkan ada post question tapi awak kena hantar lab report. Right? So lab report ni saya akan bagi masa lah bila awak kena hantar. Alright. Alright, for today's video references, okay, please look at lab number 2, qualitative test for amino acids, alright, or in any videos on YouTube. So kalau awak nak cari dalam tu, awak boleh check dalam video dalam apa tu YouTube. Klik kat situ, qualitative test for amino acid, student UiTM. So akan banyak video kat situ, right? Awak boleh tengok. Okay, today's saya akan explain mengenai laboratory manual. Alright, please open your soft copy of manual. Okay. Please open page number 7, right? For today's experiment. Right, amino acid are the building block for proteins ataupun is a monomer for proteins. Protein is a polymer. What is monomer for protein? Is amino acid. Right? They share the same acid and amino groups, but side chains is differ. When the amino acid are linked together to form an amide, the product is called peptide. So this is the bond between two amino acid. Okay, peptide is a bond between two amino acid. Or in other words, newly formed bond that is called peptide bond. Okay. If we say up to 20 are linked, they are called polypeptide. If we say more than 20 to 1,000 amino acid will link together with the peptide chain, it's very called proteins. It's a difference between polypeptide and proteins. Okay. Second paragraph is the first experiment actually. Okay. It's a experiment number 1. Okay, so this will show the expected result actually. Okay, so alpha amino acid can be detected using anhydride, which form a blue to blue violet. So the same, the actual color for anhydride is blue, but if it's positive, it will change into blue violets. Okay, other color than that, beside blue violets, yellow, orange, or red, it considered be negative result. Okay, this one, negative result. Other than that, blue violets. The test is very sensitive, often used for colorimetric determination of amino acid solution or as a visualization agent in the chromatography of amino acids. Right? So what happened during the reaction, amino acid group ataupun amino group will liberate ammonia. Alright? So this ammonia will react dengan anhydrine and giving you several color. So the positive color will be your blue violets. Other than that, it's negative. Okay? So for those amino acid yang dapatkan warna blue violet, dikenali sebagai positif. Atau dikenali sebagai alpha amino acids. Okay? So let's say awak buat eksperimen tentang makanan, so awak pakai anhydrine, lepas awak letak makanan tersebut dalam detection, tiba-tiba awak dapat warna blue violets. So maknanya makanan itu mengandungi asid amino. Tapi awak kena hancurkan dulu makanan dulu. Alright? That's the beauty because of the experiment. Alright? So, but please in mind, this one is qualitative. It's different with the quantitative. Right? So qualitative more on observation experiment, which means we have to use the measurement by using the changes of color, more on observation, right? So quantitative more on measurement, such as you need meters, centimeters. So that one is more on quantitative. Qualitative more on the changes of color, more on the observation part on science methodology, right? Okay, paragraph 3, most protein give positive result on xanthopoietic test. Okay, because presence of phenyl groups. Okay, so this phenyl group produce color nitro compound, which is react with the nitric acids, right? So this one is xanthopoietic test. Xanthoprotein test. Okay, continue. The amino acid with the thiol group, or what is thiol group? Is a SH group. For example, cysteine. Cysteine is one example of amino acid. We have to hit with the presence of alkali first to produce the sulfide ion. So this sulfide ion will detect, and with the lead cations, what we call black precipitate plumbum sulfide is formed. So maknanya kita kena letak bahan berupa plumbum. So this plumbum akan capture dengan sulfide ion. Awak akan dapat result dia. Yang mana ada result black precipitate plumbum sulfide, that one consider amino acid that have a thiol groups. Okay. That one we call it presence of cysteine. The Milan test is used to find the hydrophenyl sites on amino acids. Okay. So the color will be red in color. Okay. So we use what we call tyrosine, which is give the positive result. Okay. But however, in any of amino acid, which is have the hydrophenyl group, can be positive with the presence of red color. Okay. Walaupun kita pakai tyrosine sebagai positive control, tapi kalau ada amino acid lain yang kita gunakan, still lagi kita boleh dapat result dia. Dan kalau dapat result dengan menggunakan Milan test, maknanya amino acid tersebut ada hydroxyphenyl site. Okay. Hopkins cold test is a heterocyclic side chain which is indole chain. So heterocyclic side chain tu nama dia adalah indole chain of tryptophan red to give a purple to violet rings at the interface of two layers. Okay. So we use what we call one example amino acid yang paling mudah adalah kita pakai tryptophan. So what happen? You will get the purple to violet ring. So kenapa ring? Because you're using a test tube. Okay, between two layers. Right. The last one will be the Sakaguchi test. Ammonia or ammonium ion give a positive reaction in this test. Okay, so it means kalau kita dapat ammonia ataupun ammonium dalam eksperimen tersebut, dia akan react dengan Sakaguchi test dan mendapatkan result yang tertentu. Alright. Objective for this experiment to conduct series of qualitative tests. Okay, please bear in mind what is meaning of qualitative. Saya dah beritahu tadi. For amino acids. Okay, what type of amino acid dalam eksperimen ni adalah tyrosine, okay, tryptophan, phenylalanine, dan proline. So this one kita akan masuk selepas enzim nanti, alright? So the method number 1 in hydrate is very simple. So pipette 1 ml of glycine in the test tube, and then put a 5 drop of hydrate. Keep the test mixture in boiling water bath for 2 minutes. And record the changes. Okay, same goes to other amino acids, tryptophan, tyrosine, and so on and so forth. Test number 2, xanthopoietic test. Pipette 1 ml of glycine in test tube. Put 1 ml of concentrated nitric acid. So please bear in mind, please be aware, this is very dangerous substance, concentrated nitric acid. Swill the test tube and immerse the test tube in the cold water to cool them down. Okay, swill, all right. So record any changes, and then after that, put 40% of NaOH or sodium hydroxide solution dropwise to change the pH solution to alkaline. And then record all the changes, right? Same goes to other amino acids such as tyrosine, phenylalanine, and phenols. Okay, but phenol is not the amino acid, all right? Phenol adalah bahan, bukan amino acid. Please bear in mind. We go for the Milan test. Pipette 1 ml of glycine in a test tube. Okay. Heat the test mixture boiling for 10 minutes. Okay. Heat the test tube for 10 minutes, right? Cool it down in the room temperature and add 5 drops of nitric acid. Sorry, natrium nitric. Record the changes and repeat using stock solutions such as tyrosine and phenylalanine. We go for the test number 4, Hopkins cold test. Mix 1 ml of glycine with 1 ml of glycer acetate acid in the clean test tube. Incline the test tube. Slowly add 1 ml of concentrated sulfuric, but do not mix. So incline means you have to put your test tube in 40 degree centigrade ataupun 45 darjah dan masuk perlahan dua cecair tadi, glycine dan juga glacial acetate acid. Make sure there are 2 layers should reform. Jangan bercampur, right? That's meaning of incline. Let the layer stand and note the color at the interface after 2 or 3 minutes only. So this awak akan dapat lebih kurang 2 atau 3 minit, lepas tu baru dapat. Lepas tu dia akan hilang. So that one will be your, so check balik awak punya introduction, apa awak dapat. Okay, violet rings. Okay. For this one, you have to use a tryptophan for your positive results. Okay. Test 5, lead sulfide test. Pipette 1 ml of cysteine in the test tube. Add 1 ml of 40% NaOH. Swill the test tube. Heat the test mixture in the boiling water bath for 2 minutes. Immerse the test tube in the cool water to cool them down. Okay. And then how you capture the sulfide ion? We're using a sodium plumbate. Okay, this sodium plumbate will react with the sulfur ions. Okay, awak akan dapat particle tu, black precipitate, right? We're using another amino acid, yaitu cysteine and methionines. Okay. Last one will be your Sakaguchi test. It's very simple. Pipette 1 ml of arginine in a test tube. Add 1 ml of 40% sodium hydroxide. Add 2 drops of naphthol, 5 drops of bromine or bromine, and swill the test tube. So make sure you get the changes. All right. All right, is there any question before we continue for our actual process of chemical reaction between the test reagent with the amino acids? Okay, ada soalan sebelum saya sambung lagi? Is there any question left?
No.
Okay, so saya akan nyatakan selepas ni proses chemical reaction between the each of the tests. Okay, dalam bentuk PowerPoint right?
Okay, sir.
Okay, kita tunggu. Alright, so qualitative for amino acid. Okay, which part is biochemical process? Okay, so if you bear in mind okay, what's immunobiology, what's immunochemical? So biology is the macromolecule itself. Okay, so macromolecule tu ada macam-macam Dia ada protein, dia ada carbohydrates, dia ada lipid dan juga ada nucleic acid. Okay, so one part of the biomolecule or biochemistry tu adalah amino acid. So this amino acid is one of the biology part. What about chemical? Chemical, the reaction between amino acid with the chemical dalam eksperimen ni. That's meaning of biochemistry, alright? Okay, we go for the number one. What is amino acids? Okay, you will learn it further on the lecture next week. Okay, so amino acid is a building blocks for all proteins. Are linked acidity by peptide bond. So macam mana bentuk peptide bond? CONH to form the primary structure of proteins. Okay, so once I'm giving the lecture here, so make sure you see the structure here. This is structure of amino acid. Okay, and please check which part is carboxylic acid and which part is amino acid. Okay, poses and amide group and carboxylic acid groups and a varying side chain that differ between different amino acid. So this one is a example of what amino acids. So amide group is one of the functional group. Kalau buka di nota, apa itu amide group? This is your amide group, NH2 ni. What is carboxylic group? This is the one, COH. Okay, so this bulat ni yang besar ni represent the oxygen. So this one merah sebenarnya kalau hijau color. Okay, so this one is a side chain R ni. This one yang memberikan bahawa there's a lot of amino acid yang tahu. So only 20 amino acid ada. So tadi tu ada dengar dia ada phenylalanine, dia ada tryptophan for example, dia ada tyrosine. Okay, so that one is because of the presence of side chain. Tapi untuk structure dia semua adalah yang basic mesti ada dalam ni. Tapi R part ni adalah berbeza. That's why we have a 20 naturally occurring amino acids. Right? So what's the meaning of side chain? Side chain is this one, R ni. Okay. Okay, we go for the test. So what happened in the hydrogen test? Okay, kenapa dia memberikan warna yang berlainan? Okay, the hydrogen degrade amino acid into the aldehyde on a pH of 4, ammonia and carbon dioxide through the series of reactions. Okay. We have this anhydrine, and then we have the amino acids. Okay, so once amino acid bergabung dengan anhydrines, we get the hydrogen then tin. Okay, the reaction product obtained from the anhydrines is hydrogen then tin that react with the ammonia, NH3, and access anhydrine to produce an extensive blue and purple pigment. So what happened to hydrogen dan tin ni, dia akan bergabung juga dengan hydrogen yang dalam cair tersebut, okay, ataupun dalam test tu, membentuk lumen purple. So this lumen purple yang awak dapat tu, blue violet tu, right? The immunologic acid proline and hydroxyproline also react with hydrogen, but okay, proline is one of the example of amino acid juga. Hydroxyproline amino acid juga. Tapi berbeza dengan semua amino acid, dia akan menghasilkan warna yellow instead of purple. Tapi hanya untuk asid amino jamak proline saja. Okay. Okay, xanthoprotic acid test. The test give the positive result in amino acid carrying aromatic groups, especially in the presence of tyrosine. Okay. For example, phenylalanine, tyrosine, tryptophan, we have what we call aromatic amino acids. Okay. So in the presence of concentrated nitric acid, the aromatic phenyl rings is nitrated to give the yellow color nitro derivatives. So at alkaline pH, the color change to orange due to ionization of the phenolic group. So macam mana bentuk phenolic group tu This one structure dia. Heterocycle. Okay, heterocyclic ni. So this one example of tyrosine. So one tyrosine bergabung dengan kita punya nitric acids. You will get yellow color. Okay, because of the orange. Okay, yellow color. Okay. But in alkaline pH, kita akan dapat warna orange. Tapi kalau kita pakai electropoietic test, kita akan dapat warna yellow. Okay. Bila test, the test will test the presence of hydrophenyl site. So macam mana bentuk hydrophenyl site? So bila awak tengok structure tyrosine ni, this one, this is our hydrophenyl site group. Okay, once dia bergabung dengan HNO3 ataupun nitric acids, kita akan dapat red color. This one nampak ni color ni warna merah. Okay. So the best we using what we call tyrosine. Okay, because of what? Tyrosine have a hydrophenyl site group. Or in any molecule that have a phenolic OH we react. Hopkins cold test. Okay, Hopkins cold test reagent only react with a protein containing tryptophan. So once the tryptophan is free, it reacts with the glacial acetic acid to form violet product. Okay, so the protein solution is hydrolyzed by the concentrated sulfuric acid at the surface solution at the solution interface. Okay, so bila dapat kita hancurkan protein tersebut, dia akan terlepas bentuk seperti tryptophan, alright. So bila dia bergabung dengan kita masukkan tryptophan campur dengan formaldehyde, so tryptophan juga. So sekarang ni apa yang berlaku dekat sini, dia akan berlaku proses hydrolyze of the proteins. Okay, bila kita gabungkan dua-dua ni, condensation, kita akan dapat violet products. Okay, so the best kita pakai Hopkins cold test, kita pakai tryptophan. Yang kedua kita pakai telur. Okay, white. Itu pun boleh juga untuk dapat violet products. Okay. Alright, last one will be Sakaguchi test. Okay, so mostly we can get the red color complex, alright. But you have to create the alkaline condition. Okay, so we're using what we call alpha naphthol. So what's alpha naphthol? It's a 1-hydroxynaphthalene, which react with a mono substitute guanidine compound like arginine. So the best amino acid they use for Sakaguchi test using arginine. So it will treat with a hypobromide and hypochlorite to produce the character red color. How? So we have arginine. This is example of arginine. And then we put the sodium hydroxide. Why? Because we have to put to do the alkaline condition. And then make sure you put the alpha naphthol or 1-hydroxynaphthalene. Mix 3 distinct. Kenapa NOH? Sebab kita nak createkan alkaline condition. Naphthol supaya dia boleh capture kita punya ammonia dalam tu. Alright. Once dia mix, okay, kita akan dapat apa ni? Bromo, okay, hypobromide ataupun hypochlorite. Ni nampak ni betul ni. Ni hypobromide. Bergabung dengan sodium akan react. So hasil dari sini kita akan dapat red color complex. Red color complexes. Okay. So what about cysteine? Okay. So cysteine, one of the amino acid, they have a sulfihydryl groups. Okay. So what we get the positive result from there is a form black precipitate. Okay, this is your cysteine. So dekat sini ada SH. Alright, apa yang berlaku? Kita perlu hydrolyze this structure first. Macam mana kita boil dalam water bath. foam boiling, mostly water bath. Okay, and then what happen? This S will be putus dan menjadi S2 negatif ataupun sulfide ions. So this sulfide ion akan buat apa? Kita akan capturekan dengan plumbum PB. Okay, kita pakai sodium plumbate. So apa yang berlaku? Semua sulfide ion tersebut akan bergabung dengan plumbum dan membentuk black precipitate. So that black precipitate adalah dikenali sebagai SH group tu. Okay, so the best example using a cysteine for positive control. So ni warna dia bentuk ni. Alright. Okay, so watch video, you boleh watch video ni ataupun you boleh watch video yang saya bagi dalam kat line tu. Right. Okay, so this one PowerPoint I've already upload inside the Google YouTube future. So you can find it on the course content. Alright. Alright, is there any question so far? Ada soalan? Is there any question? All of you? About today's experiment?
No, sir.
No? Other than that? So hopefully you understand what's meaning of biochemistry. Okay.
Okay, sir.
Okay, so ada soalan lain?